So we started this project, we launched it early February 2020, when it became clear that COVID is going to be the next pandemic we are going to face, and it's going to spread all over the globe. And there were multiple questions that were unclear about this virus. For example, we did not know how SARS-CoV-2 represses host genes. Of course, it's very important to avoid the antiviral response. Also, it wasn't clear how it promotes the expression of own genes, because it's the final aim of any virus in order to proliferate. And it was completely unclear how it differentiates between own and host genes to either promote or repress. It was also a problem to start this study because very few things were known about SARS-CoV-2 in February 2020. However, it wasn't the first time the humanity faced the COVID virus. It was back to 2003 with SARS-CoV-1, and we knew that we are talking about a virus which has a genome coming as a huge positive single-stranded RNA molecule of approximately 30 kilobases. And we knew that it has a 5' cap structure and poly-A tail, so it comes ready to be translated. And we knew that there are multiple non-structural proteins, commonly termed NSPs, and we knew that particularly the first NSP, NSP1, is a major pathogenicity factor in SARS-CoV-1 because it was shown to promote degradation of host mRNAs. So we said, okay, this is a good entry point. We cloned the NSP1 of SARS-CoV-2, we expressed it in the cells and measured the half-lives, and you can see that cells expressing NSP1 have significantly lowered half-life of almost all endogenous mRNAs, suggesting that the NSP1 of SARS-CoV-2 acts similarly to the NSP1 of SARS-CoV-1. Quite interestingly, we found that spliced mRNAs are affected more strongly. You can see here that single exon mRNAs, they lose relatively few hours from the half-life, while the spliced mRNAs, multi-exon transcripts, some of them lose even 20 hours from their half-life. And indeed, if you take a reporter mRNA, and when we introduced an intron within the 5' UTR, you can see that this mRNA became much more repressed by NSP1 than exactly the same mRNA which was not spliced. However, it did not, it is a nice idea, right, because the viral mRNAs are not spliced. However, also antiviral host mRNAs, most of them are not spliced, for example, interferons. So it did not really provide us with an answer how it differentiates between host and viral mRNAs. So we were looking for an additional mechanism, and yesterday such a mechanism was presented by Oliver. Early in 2020, it was shown that the C-terminal domain of NSP1 can get incorporated into the entry channel of the ribosome. And indeed, when we expressed NSP1 in cells, you can see that there is a downregulation in the polysomal fraction. We could quantify it and see that you have a lot less polysomes versus monosomal fraction. Quite interestingly, NSP1 is completely absent from the polysomal fraction. It is mostly enriched in the free fraction and the initiating fractions. It is a bit present in the ATS, in the monosome, but is completely absent from the polysomal fraction. And like what was presented yesterday, we could perform poromycin labeling of niacin peptides, and you can see that there is a translation going on, however, it is inhibited partially. So from this, we concluded that NSP1 can interfere mainly with the initiation step, and probably it does not have a lot of impact on the translational elongation step. Quite interestingly, despite all these terrible things, NSP1 reduces half-life of mRNAs and it inhibits translation, the cells can be successfully transfected with NSP1 and they can proliferate. So you can see here 3 different cell lines transfected with either GFP or NSP1 under different 5' UTRs. You can see they grow just fine. And when we sampled these cells during the growth, you can see that the cells just don't mind expressing either GFP or different amounts of NSP1. And this is in contrast to well-known inhibitors of translation like taurine, homoharingtoning, or cyclohexamide, or puromycin. However, we found an interesting we observed an interesting thing. Here you can see we expressed NSP1 under endogenous promoter, endogenous 5' UTR, which was derived from the virus, or control 5' UTR, and you can see that when the viral 5' UTR is present, the expression is much higher. And we were able to confirm that this expression is elevated due to the differences at the protein levels, suggesting that there might be something within this 5' UTR of SARS-CoV-2 that may potentiate translation upon expression of NSP1. And we decided to go deeper into this field, into this finding, and we decided to focus on the stem and loop element. This is the first structural element within the viral RNA. It is very close to the 5' cap structure. And in case of SARS-CoV-1, it was shown to be promoting resistance to NSP1. And indeed, you can see that both genomic version of 5' UTR and subgenomic version of 5' UTR, they both include this stem and loop structure, and they both are successfully expressed when they are co-expressed with NSP1. So the first thing, we decided to play with the structure, and we decided to focus on this part of the stem and to introduce here several mutations that will open the stem structure. So you can see this is the first yellow column. You can see that the result was completely opposite from what we expected, and actually the open structure promoted the resistance to NSP1. This was quite a weird result, especially taking into account the data from SARS-CoV-1, and we decided to complement this mutation to restore the secondary structure but change the sequence. So we did it, and you can see that we basically abolished the ability of this RNA to be efficiently expressed with NSP1. So this was very strange, and we decided to remove completely the stem and loop structure and to replace it with another stem and loop structure from another virus, the chikungunya virus, which is much longer and much stronger sequence structure. And you can see that basically it did not help to the mRNA to be expressed efficiently with NSP1, suggesting that the structure is not the most important determinant in the resistance to NSP1. Next, we decided to check the role of proximity of this element to the 5' cap structure, and to do so, we introduced here a linker of 10 bases, and when we did so, we actually abolished the ability of this mRNA to be expressed. And we said, okay, this is so cool. We were very enthusiastic. Let's just confirm it by introducing here another 10 nucleotides. And when we did so, introduced here 20 nucleotides, we basically restored the ability of this mRNA to be expressed, suggesting that we quite don't really understand the role of the distance of the stem and loop structure from the 5' cap. The third thing we wanted to address was the stretch of polypyrimidines. You can see here a consecutive 7 consecutive pyrimidines. We said, okay, this is interesting. So we decided both to mutate them randomly or to shorten the stretch. And indeed, you can see that both mutagenesis and shortening the stretch reduced the ability of this protein, of this mRNA to be expressed. However, it was still quite elusive. We still did not understand the role of the viral code until this experiment. So this experiment was a game changer. And here we found a sequence which is UAUA. And now it is difficult for me to explain why we chose this sequence, probably because it reminded us, resembled the Tata box at the DNA level. So we decided to take this AUA sequence and to replace it with triple C, triple U, triple G, or triple A. And here we got a very interesting answer. Only when this sequence was replaced by triple G, the mRNA lost its ability to be efficiently translated, suggesting that the presence of guanosines actually abolishes the NSP1 resistance. Now we could take this lead and to reanalyze all our findings and to precisely map the position of guanosines. And what we saw is that it is the lack of guanosines between the positions 8 to 23 that confer the resistance to NSP1. And if you introduce even a single guanosine residue, you already lower this resistance, and if there are 2 or more guanosines, this resistance just disappears. It is so important, this precise location between position 8 to 23, that when you take a native viral 5' UTR and you just put additional 16 bases between the 5' UTR and the transcription start site, you basically completely abolish the ability of this mRNA to be expressed with NSP1. But if you do obey this rule, you are able to express very efficiently probably any mRNA, even those that have nothing to do with virus. So you can see here, we took just a reporter 5' UTR, we created artificially a stretch which doesn't have guanosines, and we co-expressed it with NSP1, and you can see that these mRNAs are very efficiently translated. So this feature, we found it to be evolutionarily conserved. You can see that multiple beta and alpha coronaviruses, they do encode for stretches, 5' cap proximal stretches that are devoid of guanosine residues. However, I told you that there are 2 different types of 5' UTRs for SARS-CoV-2. There is subgenomic 5' UTR of approximately 72, 75 bases, and there is a much longer 5' UTR of 265 bases. All of them have this stretch. So this means that the genomic 5' UTR must have some additional features. And we immediately thought that maybe it is a cap-independent translation. Now, it was a kind of naive thinking, because we know that SARS-CoV-2 genome comes capped. So it brings 5' cap, and who needs a cap? Who needs an iris if you have a cap? But anyway, we carried out this experiment. So this is a bicistronic assay experiment. I will just say that there are 2 cistrons, and if we want to check the ability to mediate cap-independent translation, we just clone here our sequence of interest. And when we cloned the 5' UTR, the long version of SARS-CoV-2, you can see that it potentiated a very efficient expression of the second cistron. Now, this efficient translation was independent of taurine treatment. So you use taurine to inhibit cap-dependent translation, and you can see that it is still efficiently translated, but it was very sensitive to flipping. So when we cloned it in an opposite configuration, we actually didn't have this effect. And in general, this behavior resembled very well the iris taken from the EMCV. So you can see that the behavior is quite similar, and we observed it in both MCF7 cells and MRC5 cells, suggesting that it is not something that is cell type specific phenomenon. However, bicistronic assay has several drawbacks, we know that. And to address that, we decided to check it in a different way. So we created several in vitro transcribed mRNAs, which were uncapped, but had poly A tail. And only when the genomic 5' UTR was cloned, it could mediate cap-independent translation within cells. But we don't care much about RNILA, right? We care about the viral protein. So what happens when we take the genomic 5' UTR from the virus and we clone it correctly upstream of the NSP1, let's say? So what happens is that the expression becomes really high, really efficient. And not only it becomes efficient, it also becomes completely independent of cap-dependent translation. You can see here, we treated cells for a long time, 24 hours, with high concentrations of taurine, 200 nanomolar, and you can see that the expression under the long version of 5' UTR completely insensitive to this treatment. On the opposite, you can see that NSP1 under control 5' UTR, first of all, it expresses in much lower amounts, and it is very sensitive to even short treatments of low concentrations of taurine, suggesting that the genomic version of the viral 5' UTR can mediate cap-independent translation. So let's wrap up. We wanted to see how COVID-2 represses host genes, and we know that NSP1 induces a degradation of host mRNAs, and it interferes with the process of translation initiation. We wanted to understand how it promotes viral genes. We know that unspliced mRNAs are less affected in terms of degradation by the NSP1, and viral mRNAs are not spliced. And we know that the genomic 5' UTR can support cap-independent translation initiation in order to translate NSPs, which are 16 different proteins. And we wanted to know how does it recognize viral mRNAs. So we know that it uses cap-proximal G-less sequences, and we know that it is the sequence and not the structure which is important in this case. So I want to thank the group of Rivka Dikste. This is Rivka from the Weizmann Institute. We performed this study in her lab. I want to particularly mention the role of Urmila and Anastasia in this study, and our collaborators, Igor Ulitsky and his student Binyamin Zuckerman, who performed bioinformatical analysis, the founders. And I just want to mention that I recently opened a new lab at the Technion. We are focusing on the RNA stress response, multiple sides of this response, and we are looking for students, if you know someone who can be interested. And I want to thank the committee for letting me stand here, and thank you very much for your attention.
All right, thank you very much. Do you have any questions?